All right, let's jump in here. So my name is Lena. I'm the Director of Education over at the Maine Tree Foundation. And before we jump into the content, wanted to, there we go, do a quick land acknowledgement. As we're going to be talking a lot about outdoor and environmental-based education today, it's really important that we acknowledge that the land that we're on is unceded territory of the Wabanaki, in most cases, if you're in Maine. And they're the original stewards of the forest that we use when we're teaching with different students outside. And so it's really important to give, to include that and to make students aware of all of their involvement and ongoing management with the land that we're on. Maine Tree Foundation is a nonprofit that is focused on environmental education and timber research. So the top 3 programs on your screen here are under our education umbrella. They're the ones that I'm in charge of. Our Forest of Maine Teachers Tour is coming up this summer, and I'll have some more information about that at the end today. And then we'll also be hearing about Project Learning Tree and Forest Ecology Research Network today as well. So if you have any questions about what we talked about today, or find anything that you think might be helpful to you or your classroom, feel free to let me know. So as we go through today, after this quick introduction, I'm going to toss things over to Katie Glover. She's from the University of Maine. And after we talk with Dr. Katie, we're then going to toss things over to Cynthia Nye, who works at Loranger Middle School in Old Orchard Beach. So, let's see. I'm going to stop sharing my screen now. And you should be all set to share, Katie.
All right, sounds good. Thank you. Just give me 1 second. Let's see, and I'm going to ask if someone could unmute or just like verbally confirm that you can see the slides all right.
Yes.
Okay, awesome. Thank you. I feel like after all this practice with teaching over Zoom the last 3 years, I still can only do like a function and a half well at one time. But it's great to be here today. Thank you, Lena, for the invitation. I think this idea started with a visit to the Massabesic Forest in January and a public tour that I had done as part of the Center for Sustainable Research or Research on Forests and the Forest Service in a public tour. And it's great to connect with you all today. And I'm pleased to be joined with Cynthia and I later in the program, who I know from teaching a professional class last summer. So I can talk a little bit about myself. I am a former high school teacher and now researcher, and now I do a lot of outreach and coordination and professional teaching for the Institute. I would like to take a moment to also recognize our land acknowledgement, which largely focuses on the Penobscot homeland and recognizes that, you know, we have this problematic past and that issues of sovereignty are still ongoing for our Indigenous colleagues and partners and citizens here. And if you'd like to read more about UMaine's efforts towards this and the memorandum of understanding that we've signed with the Penobscot Nation, I've left some resources at the bottom there. So a little bit about me. I am a former high school teacher. I taught 9th grade earth science and 11th, 12th environmental science for 6 years, kind of sandwiched between master's and PhD in graduate school. In my research, I study sediment cores from lakes and wetlands and bogs to investigate past histories of change and develop past histories of change, largely around vegetation, but also how fire impacted the landscape, how hydrology and deposition changed over time, and how climate changed over time. And what were some of the climate events or other disturbances that could have induced past change in these systems. So you can see some of the cores here in the field. This is what it looks like in the lab. If we were to take away those coring materials from that upper right picture, that's what most of my days look like now. I'm less so in front of students or out in the field and lab and at the screen because I do a lot of outreach for the Institute. Two of the big initiatives I lead right now is a new program for an online graduate certificate for professionals. And then we are about to launch, I co-lead a new working group in fire and fire history with the North Atlantic Science Exchange, and our kickoff meeting is coming up end of this month. So a lot of what I teach and how I think about my research and frame it is around this concept of ecosystem state change. What makes ecosystems move from one stable state to something new or different? And there's many examples now of course, anthropogenic change and development and how humans have started to impact ecosystems with declines in biodiversity, habitat fragmentation, that sort of thing. But I find this model is pretty accessible to most students, most audiences, pretty much any grade level. I have used it from high school to graduate students. But the idea in this visual is that we have an ecosystem that has co-evolved over centuries, maybe millennia, to have all of these processes that fit together and help it maintain this stable, mature state. But things can happen, disturbances can happen in the environment. Let's say there's a warming event or drought or there's fire. If that ecosystem is resilient, that community, that ball in the valley, it'll start to move a little bit, but it rolls back down and it stays in that state. It really takes a lot of this delta X, this work, to move it into a new state. And this terrain or the steepness of the valley is a visual signal here for resilience. The steeper the valley, the more work it takes to really pull and change it. Of course, with development and warming planet now, that topography starts to relax a little bit and maybe be less steep, and it's easier to start to change states. And then just a couple reminders about this diagram. Let's see, I mentioned the steepness, the delta work, the climate driver, the disturbance, whatever it is that helps move things to different stable states. And you may have heard other concepts like regime shifts, vegetation type conversions is another I've seen in the literature, tipping points, state changes. But at this point, I often ask students, like, give me an example or something you've observed. And they almost always have something, usually with human development or like anthropogenic activity or pollution or something like that as the driver that they mention. I did my PhD in Southern California, and one of the things I thought frequently about during that time, first, California was in deep drought from about 2012 to 2016, which was most of the time I was there studying. But we had observable in the mountains this really dramatic change in our forests. And this aerial survey started in 2010 to document just how many white pine trees were dying in the Sierra Nevada hills, foothills, and then even starting to get to the transverse ranges, the mountain ranges that are around Los Angeles. And you can see just this really visceral image here of these brown, red, dead and dying tracts of trees. This is a huge signal, like this is not resilient anymore. And sometimes we think of this model not just as one driver, but maybe it's a coupling of different things that are happening. In this case, obviously it was drought, climate is warming, and that impacted the bark beetles, which had co-evolved to have this season that really stopped about mid-October. But without that mid-October first frost anymore, they could have more life cycles and kind of grow in their population and consume more of the trees. So this is one example I thought about often as I looked for state changes in the past for the cores that I study. The other conceptual thing I want to go over today before I really start to apply this is a little bit about pollen production in the environment, especially around gymnosperms. So coniferous trees, some of the oldest trees that we have on the planet, evolved long before angiosperms did, with this strategy of having cones, seeds in the cones that get airborne, and then also pollen airborne in the environment. to germinate the seeds. And just to visualize how pine disperses, if you get spring allergies like I do, your eyes might be tearing up right now, just watching this GIF in the lower right-hand corner here. But I find this is like a very visceral concept to audiences too, of like, okay, this is where pollen comes from in the environment, and this is how it gets spread around. And pine trees, coniferous trees have evolved this strategy where they just put out inordinate number of pollen grains each spring. I want to say it's like in the hundreds of thousands per tree. You know, flowers and perennials and shrubs, their rate is a lot lower, and sometimes they've evolved to be more entomologous transport, so they rely on bees or other insects or bats to do the transportation. But conifers have evolved this strategy to be airborne and be abundant in the environment during their growing season. And this is what I've looked for in these sediment cores to do these reconstructions of vegetation change. And there's some seasonal cyclicity to this too. The pine trees and conifers and other trees, they tend to produce pollen in the spring and early summer. Grasses, they tend to do pollen production midsummer, and then you might get more like late summer, early fall, and then this wonderful thing called fall allergies from ragweed and other perennials in the environment. So again, this is pretty accessible for people to think about. I'm sure we've all had that yellow pollen on the car in the springtime and early summer. You can see a lot of, if it rains, puddles, ponds, the gutter, it starts to turn this yellow color from all of the pollen in the atmosphere. I like this picture, a colleague of mine who studies deserts, so not so much into plants, but she is standing and waiting in all of this pine pollen that was released at Lake Tahoe that week. And then same week doing a little backpacking, starting to see like these concentric rings that had formed as there was slow evaporation over a couple days. On the margin of this lake, you can see the pollen deposition. Okay. So knowing that concept, just imagine this happening over hundreds of years or thousands of years at that pristine lake that really isn't going to be disturbed. Taking sediment cores really relies on finding these small basins where there hasn't been disturbance over time. And so like millimeter by millimeter, slowly, the lake can build up its record of material that's in the environment. And it's a combination of sediment that washes in. You get like sand and silt, of course. We really like clay and silt, like little smaller particles, because that's a signal that it was a lot more quiescent in that environment and things gently settled and they weren't disturbed. You can get buildup of the algae that's in the lake too. So it gets bulked up by all of this organic content from the organisms that spend their whole life cycle in the lake as well. And then you get all the material from the environment as well that blows in or washes in, and that includes pollen. That also includes charcoal. So charcoal can get to be this really fine particulate matter, and it can be airborne too. And there's certain millimeter size thresholds of how we know, like if it's really small, like 10 millimeters, that could have blown in from a long way. But something that say like 100 millimeters probably was pretty local. It wasn't wind transported and had to drop out of a stream into the lake at some point. And so the way we recover these is just imagine having a milkshake or a slushie with a straw. If you've ever used suction to put your thumb over the top and take material out, maybe try it that way. That's essentially what we're doing with these piston cores on the left. Another strategy is a bog core, which is basically like a fancy slicer or a soil sampler. It's like half a tube of metal with a flap that you twist, and it folds around and just extracts horizontally all of the material that's there. But that tends to work a little bit better at walk-up sites and bogs. The piston coring you can do on top of ice or from boats for some of these small lakes. I'd say at this point in my research, I've probably cored maybe 100 sites all around North America, largely in Canada, the Midwest, some in New England, many on the California coast. It has been the worst thing I've ever done at times. It's been the best thing I've ever done at times. There's nothing quite like that joy of getting good recovery from a site. You see that gray clay, you know you got back to the last glacial, and like nobody's ever seen this before since it was deposited. And you're about to take it home to the lab and do the work to like reveal the secrets of what that environment was like. And we have fun too. I think we took about 15 minutes to pose this over our lunch break on the coast of California. But often there's a lot of humor with this type of work. And it's a lot of fun to introduce students to who just always maybe never got over that urge to play in the dirt or be outside. And they don't mind getting really, really dirty and wet, as I did that day in the lower left. So what do we do with the material? We zoom in, this is a photo, a really small photo of a core in sequence, like 1 meter from top to bottom. But if we zoom in and start to take small samples from it in successive layers, under the microscope, we'll start to see some of this material. There's pollen, there are spores, there's charcoal, as I've mentioned. Sometimes there are ostracods and other organisms too. The different methods depend on like how much chemicals you want to use to digest it to try and extract these different things. And here's one, we're looking at it in the field. You usually open it up, like do a quick description. Always have to remember your like top and bottom and which way is up for when you like carry it, put it in the truck, get it back to lab. You want to remember exactly which way it was oriented into the ground because that's your time sequence. And I'll point out here, this is our state agent that gave us access to the site. We frequently do this work in partnership with National Park Service or Forest Service or other state land managers and agencies that manage public lands. And working with students in the lab, I found some, they really love working with the cores and like seeing the physical material itself. Others really take to the microscope work instead and just love put on their headphones and spend hours diving down into this other world to see things no one else has seen before. And the pollen digestion is pretty intensive. We use hydrochloric acid, sulfuric acid. We use hydrofluoric acid too. So again, you know, best thing I've done with teaching and exciting students. Sometimes it's the worst with just the long days in lab and how much the chemical digestion actually takes to extract the pollen grains. All right. So we're going to like drill down really small scale now and think about anatomy of these pollen grains. I mentioned they last through all of this acid digestion. Pollen grains have this really hardy exterior to them that's made out of this material called sporopollenin. And it's one of the hardiest biological polymers that's out there. HF does nothing to it. Hydrochloric acid does nothing to it. Sulfuric acid does nothing to it. Sulfuric acid like strips the top a little bit so you can see it and identify it better, but it's one of the most chemically inert things that's out there. And that's really what helps it last in a lot of lakes to build up these histories. So that's what the exine is made out of. And this is the first of a coniferous pollen structure that you're going to see. We would call them Mickey Mouse grains sometimes. I hope that's not copyright to say that, but they have these two air bladders and that's what helps with the wind transport. That helps them stay aloft and like catch air currents. And they've evolved to have this because it's beneficial to like get as far range as transport as possible. So these are really unique and easy to see under the microscope and identify. And pine looks this way. Picea spruce looks this way too. Abies, it looks like this way too. Their size and the shape and like how the bladder attaches, there are very distinctive differences between those different trees. So you can identify between them, but they've generally got the same shape because again of that anemophilous transport. And then just a little bit more, there's a glossary of pollen terms that's like 76 pages that really goes over the specific anatomy and some of the very unique names for like the design on it and the way it's structured or its symmetry. There will be sometimes sculptured surfaces with different types of textures. There are pores that often have systematic arrangements to them too. All of these features help you identify what the pollen is. Just a quick walkthrough. Paleontologists love Latin names. I love to make sure we get the common names there too, so people can connect them to the plants that we see in the environment. Poaceae grass family is often called the Death Star pollen because it has one pore or opening or aperture on it that looks like the Death Star in space, Star Wars. Sage, I'll talk about quite a bit. Carnation, you can see just the unique shapes in like exine, how hardy the exine is, structure. Daisies are pretty spiky under the microscope. Some have deep, large pores as well. And trees, there is a pine grain, alder. You can see birch and hazel look a little similar. They've got the like triporate, the three pores, like in, 120 degrees from each other. And this is where like you build your expertise to sort of see, okay, this is a little bit thicker and the pore protrudes a little bit more and the size is a little bit different. And that's how you differentiate and identify between them. So building complexity here. Now imagine that we have slides under the microscope from like one layer and then another. And we look at those all together. This was an exercise where I had students circle all of the conifer grains. So you can see all those ones with like the body and then the two bladders. Sometimes you are looking at them from the top down. So you'll see other arrangements. So there's one that's kind of classic, but then there's a few where you see the body and then it's sort of transparent. The two bladders are tucked underneath the bottom there. And then putting that all together to like build this history over time. Pollen diagrams often look like this, where we have top of the sediment recovery of the lake all the way to the bottom. We assume it's a time sequence and that was all deposited in order, oldest on the bottom, youngest on the top. Radiocarbon dating and other methods help us determine the age for that lake. And then you take the data or the portion of different pollen from all your samples, and you can start to visualize the change that happens here. And I'm actually going to ask the audience to participate a little bit here and maybe tell me, from looking at this diagram visually, where do you think a vegetation state change happened at this lake in Vermont? Any thoughts on where there's change or differences? And I apologize, these do not have common names. Pine, you might recognize, there's different types. Alder, birch, beech. This one I'm not familiar with. Spruce, pine, oak, and hemlock are all there in order.
Well, it definitely looks like there was a decrease, and then recently, like in the last 2,000 years, is that right? A change in how much is Picea, is that spruce?
Mm-hmm.
You said?
Yep. Yeah, and I saw in the chat, Michaela said 8,000 years ago. Yeah, this lake shows really well, I think, some of the classic vegetation state and climate change that we know about, both in Maine and northern New England. But let's see what we have here. Yeah, early spruce after deglaciation, and then it doesn't really come back for another 1,500, 2,000 years. That's really common at sites in northern Maine and throughout Maine too. This one is in northern Vermont. And 8,000 years is like another, we think, linked to like climatic shift. We do know that from like about 11 to 8,000 years ago is when the solar radiation rates were the highest, like even higher than today. So that had some ramifications for plants that were able to grow and hydrology in our area too. And possibly humans too in their land use. I have colleagues at the Institute who are doing some work on these cores to tease out fire histories and if those could have been anthropogenic and indigenous burning over the last 10,000 years or so, and like where that happened and when, and if it moved around and was linked to climate. I'll point out one thing before we move on. This right here, the hemlock decline. This is now like about 5,500 years ago. This shows up at a lot of sites as well. So whenever I explore this database, Neotoma, to look for pollen histories or pollen data, I always look, say, okay, did this site have the hemlock decline too? Like, what's the farthest extent of this hemlock decline and how
Katie,
widespread
do
was
you
it?
have any ideas about what caused that sharp? That's a really sharp drop off there.
Mm-hmm. Yeah, so there's two ideas. Some papers show pretty convincingly evidence for like sudden drought. And just for some reason, the hemlock was more susceptible to it. There's another hypothesis that this was bark beetle or a pathogen. But I mean, those can happen in like a couple seasons, maybe 5 or 7 years. And that's a little bit too fine to capture in these lake records. If you're lucky, you get a sample like maybe every 30 years, and that's considered a really high resolution lake and a good lake. Often a lot of the early work that was done on palynology in Maine and northern New England was like maybe 500 years, every 500 years to get a sample. So you have this resolution mismatched. You also don't have evidence deposited in the same way that there was this explosion in an insect or pathogen. So it's really cryptic. I'm not sure, it's one hypothesis, but there really hasn't been the paleo evidence to like nail that to the tree, so to speak, to say like this is what caused the hemlock to decline so sharply. All right, and then I'm gonna switch gears real quick and talk about the site that I worked on out west. And this ended up being like kind of, again, that like pay dirt when you get a really good site. This was the longest record from a terrestrial Mediterranean region in Southern California. And it was perfectly situated in the San Bernardino Mountains to sit right between these two modern biomes. There's the Mediterranean biome on the west, and then Mojave Desert on the right. And it's linked to this picture. This is the day that we recovered forest from the site. It was 85 feet of material, about 120 years of history. And I'll drill down to like 3 of the really important key things that I consistently saw in the course of this work was pine, again, and then Artemisia, sagebrush. And I couldn't think of a good equivalent for New England and the East Coast, but this is just one of these iconic Great Basin shrubs. This also has like a lot of indigenous meaning too, burning sagebrush. It always has this really aromatic smell after rain showers, and it's used in ceremony. But if you think of the Great Basin like Utah and Nevada, little bit more moist than Mojave Desert, but this is what's often the ground cover for these areas. And we call that sagebrush steppe. All right, and those are largely the 2 communities that I think were here over time. The green here is all of the tree pollen that I counted. The orange is the non-arboreal pollen, so the perennials and the shrubs that I counted in the course of this work. And you can see there are a few dramatic state changes, especially early in the record, the amplitude and that tension between it shifting back and forth between this Mediterranean coniferous forest in the mountains to one that was more open and shrubby and like probably really resembled the Great Basin. Happens quite a bit, especially here. And I always like to point people to this, or people ask like, wow, that's a rapid crash. On this time scale, that's actually a 700-year change. And I just often like to point that out in my outreach because it looks dramatic in the past, but with that tree mortality in California today, we're seeing change happen that's like 10 years compared to 700 years before we see the impact of humans and greenhouse gases in the atmosphere. And then this is really interesting, just how much the proportion of pine really picks up late in the record. So while Maine was under ice, we do think there were a few glaciers in the San Bernardino Mountains. This would have been like its southern extent. This part of California really is the subtropics, but mountains are high enough and wet enough that there were some glaciers. But the ice, there was not an ice sheet over this lake site. And instead, we just have a lot of moisture, and the pine trees really do well. And we see this lack of charcoal, right? So coupled next to this history is the record of the charcoal that was found throughout the core. And I was really surprised to see that even though we have so much vegetation change, there's actually not a lot of exciting fire, not a lot of charcoal concentration in the environment at the time. So we kind of eliminated fire as a driver that would have moved the vegetation community from one state to the next. The charcoal in the fire really seems the most active before the last ice age. So this cooler time period in Southern California, where we consistently had pine trees and probably some fuel on the landscape. And again, the time resolution problem, we didn't quite capture some of the hydrology change, but there's probably some hydrological variability happening on decades or on centuries that's driving the fire as well. And then this all is profound to tell people because the San Bernardinos are so fire prone and at risk today. We go for thousands of years with no charcoal deposition. So fire wasn't supported in the system. And I really consider that time period from 24 to the end of the record. So 24,000 years ago to about 14,000 years ago. That's its own ecosystem state where we have the pine trees kind of released from having wildfire and being burned by wildfire so frequently, creating this community that we really don't have an analog for today in these mountains. So I showed the two pollen grains that kind of popped up really frequently to sort of construct these two vegetation states. This is what I imagine it would have looked like on the landscape. Imagine something that was close to Mono Lake today. Mono Lake is a site east of the Sierra Nevada, kind of getting close to Nevada and into that more sagebrush steppe Great Basin lake environment. It's actually a really good pollen paper that came out about Mono Lake just 2 weeks ago. They did a nice job with both pores from different parts of the lake to show that the deposition changes for the pollen, but they usually find a lot of Artemisia, sagebrush, and also giant sequoia, which is no longer in the area today. And this is the San Bernardino Mountains today. You can see there's about 7 different types of pine trees that grow there, but it's clearly under a state of stress right now. It's hard to get a good panoramic shot without seeing trees that have died from drought or bark beetle or ground level ozone. So I'm going to return to this model or concept to sort of wrap up the research part of this talk. And then I'll have a couple minutes where I'll just quickly share some resources if folks are interested in teaching this or exploring this or what's out there to think about pollen and change in past environments. So if we think back to our model of ecosystem resilience, again, that resilience, that topography of this model might change because we're under a warming climate right now. And I'm a big advocate that through this process, we need to monitor and measure biodiversity because as quantities of biodiversity change or inventory of biodiversity changes, that's a big signal that the system is less resilient if you're seeing more depauperate and less rich species in an area. One of the hopeful things towards the end of my time in graduate school was connecting with a couple researchers who worked in Yosemite National Park, and they had a plot of land that had been allowed to burn for 40 years that they were studying. And this does not look like what you expect when visiting Yosemite, right? You expect to see these woods that John Muir walked or the Sierra Club pamphlet. And instead it's a bunch of trees that have been charred. But ecological monitoring showed that that's actually incredibly diverse. There's so many shrubs, there's so many perennials now that the trees are not sucking up all the water and crowding them out. The fire has been really regenerative, and there's wetlands and standing water. This was in 2016, 2017, so at the tail end of that drought. So this is one case where having active fire back in the system helps that resilience. And now we have this site that's just amazing for browse and supporting other organisms, and a place where things can come to find water at a time where there's deep drought in the state. and we don't quite see the fire risk of just all these crowded dead trees that the state often just doesn't have the resources to cut down or manage or prevent them being a fuel source. All right, so I mentioned the pollen or the hemlock decline. This is a cool paper, switching gears to New England now. This is a good paper that I think really highlights that change. That's a compilation of 17 different lake sites with these pollen records, largely focused in southern New England. Again, like I mentioned, anytime I look for pollen data from sites in northern New England or from Maine, I always look to see, okay, at 5,500 years, does it show that decline in pseudo? But you can see a few of the sites don't really have high concentrations to begin with. It just might be their coastal location. But the sites that do have it, you can see how abundant it is. And they all decline close to that like 5,500 year time period. And then we see a few of the sites, it starts to pick up again as we get close to modern. Some of the work I'm interested in doing in the future to start developing some lake records and bog records here in New England. We've talked about the Massabesic experimental forest. This would be a great site because 80% of it burned in the 1947 fire. So being able to see that in the core and like have that as a benchmark to compare anything below it to could help develop a longer fire history. This is very close to the wildland urban interface and disturbed. It's often tough to find a good site that way. But talking to Forest Service colleagues, we've identified a few closed basins that probably were kettle bogs or from the glacial period that could be good coring sites. And then Long Pond was another project discussed for as early as this summer as just like a curiosity-driven site with a team largely led by women researchers that I'd be kind of excited to do. And that fills a gap too. All of these dots are pollen data that is in the Neotoma Paleoecological Database right now. And you might see Northern Maine had a little bit better coverage than I expected, but Vermont has one site that's publicly available now. So helping to build that record would be great. Some things I've done to teach pollen over Zoom land. I had a few exercises where I just made slides with scattered pollen grains, and we almost like role played that students were at the microscope and had to identify the different things, compare in groups, you know, look at a key. I would point people to LACCOR, or now it's called the Center for Scientific Continental Drilling. They're located at the University of Minnesota, and this is probably the largest national lab for what I do, the lake coring sites. They have some great content, including, I didn't talk much about diatoms today, but it's kind of the same concept. This is the algae that lives in the lake, and they have unique shapes and identities and species, and you can build a record of like water level or water quality over time from sediment cores. The Neotoma Paleoecological Database I mentioned, I think this is great even for grades 4 and up because you don't need sophisticated reading comprehension. You can just click around and explore and like pick different taxa that you want to investigate, different plants. You can look at charcoal, you can look at pollen. These are all of the pollen records in the contiguous United States right now. It's great for inquiry too, so students can craft a question and see like, okay, you know, what are all the sites that have a record from 5,000 years to modern in my state? And again, I approached, in prep for this talk, I approached this with, my inquiry question was like, is this site going to show the hemlock decline? And then I can pull up the record and take a look. So here's one that I explored for Northern Maine. And sure enough, if I look at hemlock, there is a decline, but it comes back a lot sooner than we see at other sites. But yeah, again, we have that 5,500 year impact even at a site as far north as Maine. So that to me signals it's pretty pervasive decline because all of those other sites in the paper were from southern New England. Neotoma links to a lot of different teaching modules and activities. I'd say there's about 15 for different grade bands. And that's supported by the, hosted at Carleton College, the, let's see, Science Education Resource Center. And I've contributed one. This was a lesson I wrote that's probably good for the high school grades and could even be used in college. It's pretty data rich. Even graduate students can dig into Neotoma, and if they do any programming or use RStudio, there's ways that Neotoma links to that, so you can quickly pull data from a lot of sites and do programming exercises as well. But this was an exploration of like just looking at what are the different sites over the past 10,000 years in California? What's the geographic differences? How did it change with climate? And then I dip into graphic design every so often. So there are a few worksheets available that I've made there to teach climatographs and just some of the precipitation gradients that exist around the state. So, and all that's here. I use G slides all the time, so I'm pretty transparent about sharing materials as well. And I'm happy to link the slides in the chat for future use, or if folks want to click on or follow up with any of the materials. Everything I just showed is linked. And then I've added just like to Popular Press, a really accessible book and a great article on just like pollen analysis and how we do this work. All right. So with that said, thank you everyone for attending today and your attention and your participation. Lena, do we want to pause for questions or should I pass it to Cynthia next?
Yeah, if anyone has any questions specifically for Katie, we can pause. And if not, we can pass things over to Cynthia.
I do have one question about whether you might make something parallel to that California lesson set for Maine now that you're here.
So good question. Give me ideas for grants that would support either a student or myself, and I'd happily put something together.
I do have an idea. Okay,
right.
Great.
Awesome. Well, we have more time for questions at the end, so why don't we toss things over to Cynthia, and we can go from there.
All right.
Thank you, everyone.
Thank you very much. All right, let's see. And need to just close up. Let's see. All right. Hello, everybody. I'm Cynthia Nye, and I teach the Gifted and Talented program for grades 3 to 8 at Loranger Memorial School in Old Orchard Beach. And I support classes at all grade levels with community science and social studies investigations. I got started in this work in about 2014 on a Forest of Maine teachers tour. And I met Dr. Katie last year at the UMaine Climate Change Workshop and took her course afterwards. So I thank you very much for the invitation to share with you a little bit about what I do with forest education. I have a few slides in the beginning that are kind of short text, and then a couple that are longer text that include like an overview of my units for the 3 projects that I'm going to talk to you about. One has to do with the American chestnut, another with Hemlock Willea-Delgid, and another with the bog system at Saco Heath. And that's where I'm looking to tie some of what I do in with what Dr. Katie has been talking about today with pollen and paleoclimatology to try and bring a little of that to my students. So you'll see a couple of places where I've tried to boost my connection to pollen and start a thread there for my students. So we have a land acknowledgement 2. This second one is new this year, as we found out that we are on the traditional land of the first people of Old Orchard Beach, the Pequawket Abenaki, and we honor them and the land. For me, teaching is about environmental literacy. I want my students to learn to read the world around them, to work in the woods, and to be positive factors in their local ecosystems. I think that science and social studies are the reasons that kids learn to read and write and do math. And I found that science work in particular is very inspiring for them. I start out with a series of foundational lessons, and then other teachers have used these too. And I'm sort of thinking this as seeing the forest and the trees, kind of getting kids familiar with the forest around them. We're at the beach. So I start with something very familiar to them and ask them to identify factors in this ecosystem. And then I sort them by A and B, and then share with them that A is abiotic and B is biotic. And we get a common vocabulary going there. And let me see if I can get back to my slideshow here. And then we do nature observations with this, it's very touchy. This four-corner organizer. And I've had students as young as third grade be very excited about recording the details that they observe in an ecosystem. This one is for a naturalized pollinator buffer against our school woods. And they look for biotic factors separated into plants and animals, abiotic factors, and human impact. And then I created a tree leaf identification for just the most common trees, trying to teach them about discerning between evergreen and deciduous trees and identifying the most common in our specific woods. And then the color photo there is of one of my older students leading some third graders on a woods walk at our teaching forest at Milliken Mills Woods. This was a donation to the town, and the Conservation Commission and the school have worked together to make it a teaching forest. So we have tree identification signs. We have labels for vernal pools and for edible plants. Students over the years have put in some posters, informational posters on this old building that's there on the property. High school students built this fence around the gravesite of a Revolutionary War veteran. And this photo shows older students and younger students looking, I think it's wintergreen that they're looking at. So this property, we've learned about the Abenaki and lumber mill history of this property, and now it's a conservation and education area. And we always talk with the kids about how can we help conserve this piece of property, this land, what's special about it, and educate the public. Then we really get up close to the American chestnut. We have American chestnut at the Milliken Mills Forest, and it all has blight. And so the students see over years, as we visit again and again, they see how the blight is impacting the trees there. A few years ago, is there a way that I can close up this so that you're not, are you seeing my screen, or are you seeing all of you on my screen as well?
Cynthia, we see your screen desktop, but not Zoom people.
Oh, not Zoom people. Okay. So I'm seeing Zoom people too, but I'm glad to know that you're mostly seeing the desktop. Okay. And then over on the far right here, 2 years ago, I found a mature American chestnut off the Eastern Trail on the edge of Old Orchard Beach on the border between Old Orchard and Saco. And at the time it was dropping these burrs. And then it also had the stamen on the trail. And when I called the American Chestnut Foundation to register this tree, if it wasn't already, which it was not, they introduced me to Professor Tom Clack at UNE. And so I've really built some units, some lessons around the American chestnut. One of the things we do is we gather the leaves and then the kids, we make bookmarks. And so the kids have these bookmarks of the American chestnut leaves. We go to visit Tom Clack's American Chestnut Nursery at UNE. He is working on getting approval to plant in the wild these genetically modified American chestnuts that have one wheat gene to resist the blight. So in this series of photos, he is collecting pollen from one of those trees. It's only 1 year old. They're able to grow them very quickly in this specialized nursery. And then my students are putting some of that pollen on slides to look at it under the microscope. We do teaching around this with a lot of resources, video resources that Professor Clack has shared with me to use in the classroom. And then in the spring, we went to the research orchard in Cape Elizabeth, and students helped plant some of the seedlings that they had seen in the greenhouse. He talks to them about mycelium, this red oak mycelium that he collects and that we put into the holes when they're planting the trees. And they learn to do it there. And then we grew non-GM, so we grew native chestnut seeds. I bought this grow light and the kids figured out how to make a PVC framework. And then we planted the seeds, and some of them were from the Viles Arboretum and some of them were from a tree in Buxton. And most of them sprouted. And then we were able to plant them at some recreational department property. And the Conservation Commission and community gardeners came out and helped the students plant. Then we visited Sweet Cream Dairy in Biddeford, which makes American chestnut ice cream, which was a lot of fun to go down and talk with them, with the man who makes the ice cream, and to eat some of it. My students read the USDA research findings from a couple of years of testing of this genetically modified chestnut with the one wheat gene. And out of that reading and research, the students wrote a comment requesting or supporting approval to plant these GM chestnuts in their native range to benefit species, including people, and to boost carbon sequestration to combat climate change. And this on regulations.gov, this is their comment. They wrote it together, and then I posted it for them. And when I put it in, it kind of lost its formatting, but the information is all in there, and I think they did a good job with it. I apologize that this is jumping around so much. I'm not exactly sure how to fix that. So another ongoing investigation we have that started in 2016 is Hemlock Woolly adelgid. We went to Ferry Beach State Park to see what a heavy infestation looked like and the impacts it was having, and the hemlocks just of all ages are dead or dying there from this heavy infestation. And then we use an HWA investigation from the Gulf of Maine Research Institute. I do quite a lot of collaboration with Gulf of Maine Research Institute, or GMRI, and I've used many of their ecosystem investigation modules. So this is Project Hemlock Woolly adelgid, comes with a teacher's guide, learning outcomes. This one can be used with grades 3 to 4 or 5 to 8. And it, I think, is a series of 6 units that includes 6 like sessions that include a protocol for gathering data. You check 10 branches on the tree looking for HWA, and then you do 10 trees as a group so that you have 100 branches, and use, if you have 1 or 2 branches that have HWA, that's a light infestation. If you find many, so sometimes we found as many as 90 out of 100 branches with HWA. So we know some of our forests have very heavy infestations. There are now more than 40 schools and organizations involved in collecting that HWA data in Maine and New Hampshire. And in a GMRI webinar, we met Colleen Tierling, who's a state forest entomologist, and she told the group about a predator beetle. And this is the adult predator beetle, and this is the larva attacking an HWA egg sac. So these beetles, there is one type that's native to North America, and New Hampshire is experimenting with them. But in Maine, we have been using this beetle, which is not native to North America, but does die. It's not adapted to eating anything except HWA. So Maine has approved this beetle for use in our hemlock woods. These green slides are from a presentation that my students put together of their experience acquiring the beetles and releasing them with help from Colleen that they shared with GMRI just last week. So that's why these slides look a little different from my slides. Colleen Tierling came to school and she showed the students how to release that. She came twice, once to help identify trees that would be good for releasing the beetles on, and another time when in June to release the beetles. So I ordered the beetles from a website called treesavers.com, which is a lab that grows these beetles. They cost about $2 a piece. So students wrote to the Conservation Commission and got $500 to buy 250 beetles. And we released them in the woods behind the school. And the Ocean Park Association, Ocean Park is like part of Old Orchard Beach. They learned about the project and they released some beetles in their park. And now both of those conservation groups are researching the use of pesticides along with beetles to have an integrated pest management system to try to preserve the hemlocks in our forests. Going forward, we'll be able to release the beetles on our own and just report the locations to Colleen. And let's see. We also do a lot of projects around pollinators. We started in 2019 with a third grade community project, and students researched pollinators and created these posters that are on display now at the Milliken Trail parking area. And they planted, with the help, the Conservation Commission built them a raised bed and helped them purchase the plants. So the Conservation Commission paid for the plants. The students picked the plants to put in here, and students planted this garden. And then in spring of 2022, a colleague and I partnered with a community group in Old Orchard called Community Friendly Connection, and with librarians at our public library. And we got a grant from GMRI to focus on pollinators and climate change and to build pollinator gardens. All of those collaborators were working on pollen projects separately. So we joined forces to get this grant and use it to kind of coordinate and integrate our efforts. So far, we have planted 26 pollinator shrubs around the tennis court to help with erosion mitigation here. And these are some of my students doing soil tests at that area. This student is taking photos of bees at our naturalized pollinator planting area from several years ago. And there's now a website, Bees of Maine, and students were contributing their photos to that, so that and then sharing information with students who were doing the same unit in other schools. And then the art teacher, I had a collection of dead bees and gave those to her along with some magnifying lenses, and she did a unit with students on scientific and observational drawings of bees. Our partner Community Friendly Connection ran a pollinator art contest and used the winning entry. There were 31 entries, and they were on display at the library, and the public voted on them. And then this winning design was used to make signs for all the gardens that Community Friendly plans to install around town. This is our part of our fifth grade. The entire fifth grade participated in the planting and participated in the blueberries, bees, and climate change unit that GMRI developed, and we were field testing last year. And then this boy in the bottom right is using Phragmites stalks to make a bee house, and other students use knotweed. So we have those. We do a lot with invasive species, and students learn about Phragmites and knotweed. And so this was a kind of a cool way to bring those units together. Then, as one of their choice projects at the end of the unit, 2 students wrote to our state representative, Lori Gramlich, proposing that we have a state butterfly to help raise awareness about pollinators. And staff from the Maine Department of Conservation, Agriculture, and Forestry came to school, and they did a wonderful presentation to teach students more about pollinators. And they recommended 5 butterflies to consider. And the students chose this one, the pink-edged sulfur, which lays its eggs on blueberries and can be seen in lots of areas around Maine. And that bill is going to be heard in April. So we try really hard to help students feel equipped to speak up for the forest and for the various ecosystems around us, and to take some action in the community, whether it's educating younger students or educating the public, or in this case, writing to their state representative. That butterfly proposal started with the Hessel's hair streak, which the students learned about at Saco Heath. That's a very cool bog system that we have. We take all our 5th graders there, and they do a scavenger hunt. and they look for the unique species that are there, which include, for trees, include tamarack or American larch, which are deciduous conifers, kind of an unusual tree. And then this student is meeting a Atlantic white cedar, which is the host tree for that Hessel's hairstreak butterfly. And I want to connect, when I was taking the course with Dr. Katie last summer, she was talking about the bog pollen, and she shared an activity with me that I want to use with my older students that will create a model of the layers of a bog core. And I'll get to the end of this and share a picture of that from a link. And then she also shared an article about the Saco Heath and the paleoclimate connections there. And I want to excerpt that for my kids so that they see that this area that they get to investigate and experience is of interest to scientists, as well as to fifth graders. And then my students take a closer look at the bog ecosystem. They want to go there in all seasons, so I took them in the winter on snowshoes. And this sign talks about a plant that's 1,000 years old that's in the bog. And this student is looking down at the sphagnum moss and seeing how it's different in the winter from how it was in the fall. And we noticed that the tamarack trees have lost their needles, but they have these cute little cones. So it will be fun to go back in the spring and see the new needles coming up on the tamarack trees. When we do those bog pollen lessons, and I'm hoping in the next couple of weeks we'll be doing those, then they'll be learning more about climate change impacts on both bog and forest ecosystem. And as I was thinking about what we do with the hemlocks and with American chestnut, I was thinking every area has a forest, and people, if you're as a teacher, look for your own special forest area and partner with your local land trust or conservation group to explore and preserve it. We've also had really good connections with our soil and water district, both with Cumberland County and York County. Audubon, we've worked with them. There are lots of organizations that are really happy and looking for teachers to partner with and to work with students. So if you don't have chestnuts or hemlocks, ash, elm, maple, pine and spruce, they're all important. They all face threats. They can all be connected to pollen, the research that Dr. Katie's doing. And GMRI has ecosystem investigation modules. The bog pollen activity that I'll show you has a lake pollen option. And I have found that all the units that GMRI puts together, that 4-H provides, and those SciEd links for the pollen activities, they all include next generation science standards and usually Common Core standards for connecting to like data, the data standards for math and communication and speaking and listening, science reading and writing from the Common Core. And these are some related activities. This 4-H has a dissect a flower activity. That's a lot of fun to do. So I linked that in here. Very self-explanatory, easy to do, and really a lot of fun for kids. And I did it as an adult and I really enjoyed it too. Biodiversity Jenga is a pretty cool activity. After we did, what the Jenga towers are, with the tumbling blocks. And one of my kids said, this was the best day of school ever, just because they got to play it. They love to play this. GMRI has a vernal pool investigation, an invasive plants ecosystem investigation, and a tick investigation. A lot of students are very afraid of going into the woods because of ticks, and getting them familiar with ticks and looking for ticks and bringing them back and looking at them under a microscope, I think helps to allay some of that fear that they have. Findings from the field is a middle school journal from GMRI. Students can write science articles or nature notes and submit them to findings from the field. This is the June 2022 issue. My students have 2 articles right at the top here, but there are lots of articles submitted from students around the state of Maine. And so you can have your students read some of these articles and findings to see what other students are noticing in their area. And it all connects to sharing data and sharing findings. And communicating your findings. I use the Maine climate plan, Maine Won't Wait, for kind of our 7th grade team. Our 7th grade science teacher does a trimester long unit on climate change, but it's global and general. And so with my students, I can make it more local and focus on Maine climate impacts. My students created a trail map for all the trails in Old Orchard Beach and talked, oops, I am sorry about that. Shouldn't be on there. I wish I could show it to you. It was a very nice little trail map that they put together with some art on the front, and then information about all the different trails in town and what kinds of species are found in different trail and conservation areas. And this was a pamphlet that a student did. It folds into 3 parts about very common invasive plant species that we have in Old Orchard. I was thinking there are different projects that students do after we do these units from GMRI, so that they are finding some way to share what they've learned. My students love mushrooms, and so I just made a slideshow where they can share their photos with each other. And then I really want to do the fern plot that Maine Tree has. It used to be called a fig plot, the forest ecology research, I think, is it network. I want to do that in the next year or so with my students, to start one of those. These are my Maine Tree connections. One thing that I learned about when I was out in the woods with Maine Tree was having Maine products and Wabanaki baskets and things like that in the classroom for a quick reference. So I have that, like when we're talking about volume for math, or ash trees and emerald ash borer in science, Wabanaki arts for humanities. I have those products in the classroom so that kids can see quickly what we're talking about. And there's a nice connection with maple syrup. I buy the Arnold Farm maple syrup, but I also have a couple of students who are in Boy Scouts, and our Boy Scouts tap trees and boil syrup and then have a pancake breakfast. So I have, and they have brought information about Maine maple trees and products into the classroom too. I linked in a lot of resources, a mix of units that I use and work from my students, and this iNaturalist Bees website. So most of the things that I've talked about here are things that you could look at again and adapt for your classroom. This, just step into this paleo climate and pollen activity and show you the picture of, tells you how to set this up, but this is what you'd be creating in a graduated cylinder, layers of soil with different, they're just paper hole punch of different colors that get mixed in to represent pollen from different species of trees and grasses. And then students separate them. They look at what's in like a sample from each layer and try to figure out and map what trees and other species were in the environment in each of those layers. And I haven't done it yet, but I've mapped it out and it looks like it's going to be a lot of fun. So, and finally, in the end, I just put in a link for the native land if you wanted to find your own, the people who were originally the caretakers of whatever land your school is on. And I thank you for listening and hope you have many happy hours learning and teaching about Maine's forest. It has really made my teaching a joy, and to see the kids responding to being out in the environment, whether it's out on the trails or working on things in the classroom to learn more about their ecosystems. It's really very exciting and rewarding work. So I would welcome any questions. I'm happy to share anything that we've done. I have worked very closely with 3rd grade, 5th grade, 7th grade, and I have my own 3rd through 8th grade groups. So that range, I feel like I can be a resource if anybody is interested. So I'll stop sharing, and thank you very much for your time and patience.
I did see one question for you. Is there a way for current students to support the state butterfly initiative that you guys started?
It's being proposed. So Lori Gramlich is going to propose the bill in committee on April 4th. And then I think it may be open for public comment. But if somebody's interested in following up, yes, I'd be happy to get back, like report back and find out. I don't think we have 2 students who will be helping, like sharing the letter that they wrote to Lori Gramlich. And then the rest of the students are in the gallery. But I think it's a committee hearing first, and then it goes through approval by them, and then it goes before the full house. So I will.
Awesome.
Who is asking that question? And it's okay. Yeah. So
I thought it would be a good, if there was a letter writing campaign started, a good short postcard from each student after they
that
do.
could be really cool. I don't know. The people from the Conservation, Agriculture, and Forestry thought there was a really good chance that this would just sail through, because about 26, I think, states have a state butterfly and Maine doesn't. And it's sort of like a fun thing for the legislators to vote on. But I'll definitely let you know if there's a public comment request. That certainly is happening for the American chestnut, that it's open nationally for seeking public comment. It may be closed now because they're going to be voting, and hopefully the approval will come through in June or July. But yeah, it's a really good reason for kids to write. They're very responsive about that. So.
Awesome. So I am going to share just a few more slides here, and then we'll have some more time for questions. So if you have any ruminating, feel free to put them in the chat while we're just jumping through the last slides here. So for everybody that, if this is your first Window to the Woods session that you've done, you'll be getting access code from me for this guide. And so I just wanted to point out some of the activities in here that relate to what we talked about today. This upcoming season in the transition from winter to spring for me is like my favorite time to be out in the woods with students. And doing activities like bursting buds, where you really take a close look at tree buds and the process of creating leaves, and why they do that, and all of those different aspects is really fun. But another really cool way that relates a lot to pollen is through this activity, Have Seeds Will Travel. For the younger kiddos, it kind of introduces them to just the topic that trees have seeds and have different ways of getting those seeds out into the world. And so they sort them and look at different methods of seed dispersal, and can be good setup to talk about pollen later on. One of my favorites for grade 3 through 5, we have so many different options from Cynthia, but I also really love tying things together at the end. If you have a couple different opportunities to take some different aspects and look into certain aspects like hemlock woolly deligid, for example, if you have a couple different aspects of a forest that you're looking at, tying it all together through like a web of life activity can be really fun. Just like with the biodiversity Jenga she mentioned, kind of showing that it all trickles down, right? And then, of course, if we're going to dive into looking at how a tree is impacted by an invasive species, or maybe not an invasive species, but a disease, looking at the different parts of a tree can also be super helpful before or after, to just kind of dive in and say, like, this impacts it, but what does that really mean? And especially, like, what would that mean if it were a human kind of comparison? Talking about like tumors on a tree, and how the tumors on a tree don't move, but in humans, our cells are moving because we're moving. And so cancers can move in human bodies. Those types of comparisons can be really interesting for kids to make. And then there are a bunch of other options too for middle school and high school. One of my favorites, especially that ties into Katie's research, is looking at succession and that process of change of an ecosystem over time. A lot of times you're looking at it at a much shorter time scale, but you can look at something, a disturbance like a fire, or in Maine specifically, you can look at a disturbance like a forest that might be taken down by a good amount of beavers, or porcupines can really do some damage and really create this opportunity for an ecosystem to kind of recreate and start over from a different point. And so that's also one of my favorite things to talk about, and has some really great tie-ins to natural history, because you can look at the different phases of a landscape over time. So we also mentioned maple, and I would be remiss if I didn't talk about it at this time of year, because maple is such a great teaching opportunity in the spring. There are a bunch of different tie-ins for maple, even just discovering that, like, what a sugar bush might be, or what that a maple forest might be, and how that kind of connects to something that we use really frequently. But there are so many different ways to dive in deeper. I mean, the process of sap flow is very complex, but you can kind of simplify it and complicate things if that's something you're interested in. But also discovering and looking at the shift of the maple sap season with temperature change, and how different winters might impact the local people that have businesses around maple, is also a really fun way to incorporate it. I will say those folks are amazing to bring into the classroom. This is a very busy time of year for them, so you might want to wait until like April or May if you're going to bring in a local sap producer. But they do really love to bring in samples, so I'm sure there are some local ones that we can connect you to. And we also mentioned fern in there. So the Forest Ecology Research Network is one of our education programs. It's a field-based exploratory citizen or community science program. So on the right, the map here are all of the existing firm plots that are giving us data. There are a bunch more out there that just have the data for themselves and do stuff with it in the classroom or the community. But these folks are reporting data back to us so that we have like a statewide database of these 1/10 of an acre plots across the state. And it's just looking at the species diversity of trees on that plot, like their size, an estimation of their height, and then looking at how that changes year to year. So it looks really complex when you look at it this way, but it breaks down into something that's really quite simple. And there are also some really amazing community resources we have, like Maine's district foresters who can come out and set up the plot with you or for you. They also bring all of their fancy forestry tools and show students the process of how to use them. And there's a lot of math that they can dive into too, if you want that. They don't have to, but they are very good at talking about diameter because diameter is so important for foresters and such. But for the main portion of Fern, the biggest aspect of it is to just look at the tree species that are over 5 inches in diameter and kind of recreating what that forest looks like in a data format, so that it can be tracked over time. So you number the trees with little tags so that they can be measured year to year. But as we know, trees grow pretty slowly. So if you're doing annual data collection, it could be a little anticlimactic. So we have other aspects of the project too that look at the smaller trees like the saplings, and the understory and the dead material are some of my favorites because it looks at trees that might have been in the plot or measured a few years ago or even last year and have died in the meantime, and what that kind of means for the ecosystem and how that's actually a new resource for different species to tap into. So in your guides, I wanted to make sure that you know, so the project learning tree guide I talked about earlier is all standards aligned. So I don't think I have a guide right here, but in the back of each activity, it has a section on standards that you can find there. But there's also a whole PDF that dives into all the Common Core and NGSS and all of those different things for every activity and every grade level. So I can get that to you if that's something that's helpful. And then there are some really great indices. I love a good indice in the back of the book. It goes by different subjects, and they also have another index that has topics that you can go through. So pollination might be one in there. I know there's pollution and conservation, community tie-ins, those types of things. If you have something that as it relates there, you can kind of look at the activities by grade level and see which ones might be of interest and jump from there. And this is what the standards look like for that. So very last thing, I just wanted you all to know that our Forest Maine Teachers Tours are coming up this summer. We are very excited for them. It's one of the biggest events we do all year. There are 4-day immersive tours through the forest product sector. So we do 2 different tours. We're doing one in the Katahdin region, and then another in the Down East region. So we bring teachers out to, we go to a bunch of different sites. We see what a cut looks like, and all the different equipment that's used and the forest management plans that are created, and how that's all managed and monitored. We go to a mill, some sort of forest product mill, and watch how the trees get turned into a product. And then we also take a bunch of different stops along at some fun, like more or fun products, like less lumber and more. Sometimes we go to maple sap. Last year we went to like a snowboard making company that makes them out of Maine trees
Cool.
and stuff. So there's a lot of really great things that we covered there. And you get to meet a lot of natural resource professionals who are really excited about working with classrooms and getting students exposed to all these different opportunities they have in Maine forests. So that's here. If you would like that, I can answer any questions you might have on that. But with that, I think we can just turn it over to any other questions we might have. So I will, just so everybody knows, I will be sending out the recording and all the different resources we talked about today out by the end of this week. So you should be getting that, and you'll also get the access code if you don't already have one as well. But if we don't have any other questions, thank you so much for coming today. And feel free to let me know if you come upon questions or have ideas on how you might want to implement things in your classroom or whatnot. Happy to answer those as we go along.
Thank you for putting all this together. I
Yeah,
really
thank you
loved
for
being
sharing.
Yeah,
part of it.
same. Great to see everyone. And Cynthia, I
Yeah.
just wish I had been in your class at that age.
Thank you very much.
I liked being in your class. It made a big
Thanks.
impact on me.
Awesome.
Can teachers go on a second tour?
Mhm.
Okay.
Yeah.
That'll be
Yep.
fun.
So it's the same process. I think there's a question that asks you about that. But otherwise it's the same process.
Cool.
Awesome.
Right.
Well, I hope you all
Sounds
enjoy the
good.
rest of your night.
Yeah.
And don't lose power.
Snow is whirling now. It really picked up in the last
Yeah.
90 minutes.
For
So
sure.
I'm gonna think about whether I want to preemptively clean off my car or not.
Oh,
Yeah. Take care,
we had a
everyone.
remote day, so we got to be home. Working from home, I mean, teaching from home, but at least not driving, and no school buses trying to make it safely around town.
Mm.
All right. Well,
All right.
thank you, everyone.
Yeah.
Yeah,
Thanks
have a
again,
great
Lena.
night.
Take care.